Okay, so far we have learned about enhancers and how it can activate transcription, etc. Now we will get into a little bit molecular and structural detail about transcription factors and how enhancers actually function. So let us first start with transcription factors and how they interact with the DNA. If you remember in one of the classes, I talked about that how the major groove is considered as the second genetic code. Like these transcription factors, they actually bind to the major groove of the DNA. And in this major groove, the particular sequence that it projects makes a whole lot of difference in terms of which transcription factor it will bind to. And that's where the sequence specificity of transcription factor binding comes in. Now, what recognizes this sequence? The sequence recognition motif is typically an alpha helix that will make contact in the major groove. So what you are looking at is a bacteriophage repressor, which this green part is called the recognition motif, and that makes contact with the major groove, and some contact in the minor groove is also reported, but majority it is the major groove contact. And if you look at it, these depressors are typically bound to two major groups right next to each other, so they actually make contact among them as well. So there is some cooperativity in terms of protein-protein interaction, which helps the protein-DNA interaction. Okay. Now, this discovery in bacteriophage repressor actually helped in us understanding about the DNA protein interaction in the context of eukaryotes as well, because the operating principle remain more or less the same. So I'll quickly take you through some of the typical structural motifs that are involved in the transcription factors. One very common one is called zinc finger motif. So the zinc finger there is nothing that around a zinc ion, few amino acids organize themselves in a slight protrusion-like structures, which is an alpha helix, and that is what is called a zinc finger motif. And there are two kinds of zinc finger motifs, like C2H2 or C4. C2H2 is basically two cysteines and two histidines, and they coordinate to a zinc ion and forms the zinc finger motif. Or in C4, C4 this is just four cysteine residues. So whatever it is, because of the zinc ion, the amino acids organize themselves around the zinc ion to form a protrusion-like structure, which is an alpha helix that makes contact to the major groove of the DNA, and those are the zinc finger transcription factors. Many such transcription factors are there. If I am not mistaken, one of my favorite ones, GATA3 is a zinc finger transcription factor. The other one that is there on this slide is the leucine zipper transcription factors. Okay, the leucine zipper, as the name suggests, that there is a zipper-like part here. I will elaborate it in the next few slides. So if you take a typical leucine zipper transcription factor like this one called FOS. Okay, you will see that there are these hydrophobic amino acids like leucine, one, then after four amino acids, another one, then after seven amino acids, another one, then again after seven amino acids, another one. Like this, you will see this recurring pattern of appearance of a hydrophobic molecule. Now, if you think about it, that in every turn, between two successive amino acids, there is a 110-degree rotation that happens. So around 3.5 amino acids, there will be a complete, so the every seventh amino acid will be on the same phase as the first one. Okay, simply because of this rotation that is happening from one amino acid to the next, to the next, to the next. So you can, this is just to show you the sequence from the highlighted part, and you can count from here how the leucine residues are. This is what is the source of the name called Lucid Zipper. And you know, like if you draw what is called a helical wheel projection of this alpha helix, then you will get to see it like this. So follow me carefully. What is a helical wheel projection? What is a helical wheel projection is that you are looking at an alpha helix from along its axis, okay, looking down its axis. So what will you see? If there is a lucid residue here, number one here, then the second one will be 110 degree away from that. So this is 90, 110 degree away from that. The third one will be another 110 degree away from that. So this is here. The fourth one will be another 110 degree away from that. The fifth one will be here. The sixth one will be here. And the seventh one will be here. Okay. So now you can see that this is 7, right? So now you can see that starting from the first leucine is number 4, the first leucine, the number 4 and number 7. If this is a helix, like you imagine a 3D projection coming out of it, these two will be on the same face of the helix. Okay, so if you think of a cylinder like this, all the hydrophobic residues will be on one side. Okay, and if you have two such helices, then these two hydrophobic surfaces, they will have a tendency to exclude water from within. So they will interact with each other, which is called the hydrophobic interaction. And that's how they kind of zipper against each other. And that's why it is called a Lucid zipper. Okay. So this is the helical wheel diagram for a Lucid zipper molecule. You can see how all these hydrophobic residues, in this case it is leucine, but in some cases it can be other amino acids which are also hydrophobic. So they all arrange along the same face of the cylinder or the helix, and that's how they interact with each other through hydrophobic interaction. And then they will bind to the DNA in the major group. They pretty much have a scissor-like action on the DNA double helix. Sometimes the residues that directly contact the DNA, they will be basic amino acids, like positively charged amino acids. So therefore, they are called basic leucine zippers. So these are called B-zip proteins. These are very common motif of transcription factors. These are very common type of transcription factors. And like this is how the Phos and Jun is a very common pair of transcription factors. These are particularly involved in immunology, the genes that are involved in immune response, regulating of those genes. So here, through the green fluorescence, the hydrophobic amino acids are shown, that how they form the zipper, and this is where the basic domains are actually interacting with the DNA in their major group. So this is how the Lucid zipper molecules work. Then there are other kinds of transcription factors like Krebs. Okay, Krebs is a transcription factor which is like a cyclic AMP response element binding protein. So when the cellular glucose level goes down, Krebs gets activated. Okay, it gets phosphorylated, and this phosphorylated Krebs is basically structurally, intrinsically, it is unorganized. It does not really have a good secondary structure. But then when it comes in contact with Krebs binding protein or CBP, due to this protein-protein interaction, you can think of it in the context of induced fit model. So what happens is that when two different proteins are coming together, then you will form a new set of hydrogen bonding interactions, hydrophobic interactions, ionic interactions. So that will cause some structural changes in both the protein components. The structural change that happens in the activation domain of the transcription factor, in this case Krebs activation domain, it will undergo a transition from relatively unorganized structure to an organized structure, and then it will interact with the DNA major group, it will interact with the other transcription factors or the mediator complex, and then interacting with transcription. A slightly different scenario is there with the nuclear hormone receptors. In nuclear hormone receptors, which are transcription factors, they are called nuclear hormone receptors because they interact with these small molecule ligands, which are acting like hormones, like testosterone, estrogen, vitamin D3, etc. So these nuclear hormone receptors generally are present in the cytosol. Upon binding to the ligand, it moves to the nucleus, and there it interacts with a co-activator. Okay, so when the co-activator is present, here again the co-activator will generally have a like a globular structure, and then when the ligand bound activation domain of the estrogen receptor interacts with the co-activator, then it assumes alpha helical conformation. It's slightly different, but the bottom line remains the same. That in many cases, when there is an activation domain that interacts with the co-activator, there will be structural changes in either of the proteins or both the proteins, and that is necessary for the transcriptional activation. The reason it is important to have this kind of a control is that it is not a single factor dependent turning on of transcription. Rather, the presence of co-activator is needed for the transcription to be turned on, so that the dependence is not just production of Krebs or estrogen receptor, but some other factors which will result in production of the co-activator as well. And you will be able to understand this a little better in the next 2-3 slides. So now you think about what is happening in an enhancer. So as we said, that an enhancer is a stretch of DNA where multiple transcription factors can bind. Okay? Many of the transcription factors, like whether it is the basic helix loop helix transcription factors, or homeodomain transcription factors, or B-zip transcription factor, whatever it is, they typically bind as a dimer. Now they can form homodimers, they can also form heterodimers. And because they can form heterodimers with a variety of interacting partners, so immediately the number of combinations of transcription factors that can activate a transcription goes up dramatically, right? Because if there are 1400 transcription factors, and if you imagine that a particular transcription factor can heterodimerize with 20 of them, then the number of possibilities go up significantly. And each of these will have slight variation in their activation domain or the repressor domain, and that will slightly modulate the rate of transcription in one way or the other, right? Because depending on which particular combination, the surface that it is offering to the mediator complex will be slightly different, and that generally does change the rate of transcription, right? So, what is shown here is that these are all, homodimers of three transcription factors. These are heterodimers, okay? Now, the there can be that the binding site for two heterodimers are more or less the same. But it can also be that the binding sites for each of the monomers is completely different from each other. Okay? Look at the color coding. So therefore, and in some cases, what can happen that the particular transcription factor can also interact with the repressor, which will not allow it to bind to the particular the DNA sequence for this transcription factor. So when you bring all of this together, then you can realize that this interplay of many transcription factors, some of which are activators, some of which are repressors, can give a very different character to a particular enhancer. So, and that affects rate of transcription in multiple different ways. A very good case in point is NFAT and AP1. NFAT and AP1 are two transcription factors that are critically needed for transcription of a gene called IL-2. IL-2 is a critical component of your immune response. Okay, so now overproduction of IL-2 can cause pathological conditions, including rheumatoid arthritis or this kind of autoimmune disorders. Now you think about it. It's a very classic case. NFAT by itself has a very low affinity towards the DNA. AP1 by itself has a very low affinity towards the DNA. However, if NFAT and AP1 both are present and their binding sites on the particular DNA are close by, then they physically interact with each other at the protein level, and then they also bind to the DNA, and then it is a much more stable complex. So try to think about it another time. So these two proteins, they have physical interaction, protein-protein interaction. They both interact with the DNA, but anchoring them to the DNA facilitates their protein-protein interaction. Therefore, like these two proteins form a stable complex, which in turn reinforces their binding to the DNA. So everything taken together, you get a very stable complex formation. Now, how does it help? Because the upstream signaling pathway that leads to production of NFAT is one, and the upstream signaling pathway that leads to production of AP1 is another. So if both of these upstream events are happening, then only IL-2 transcription will happen. So therefore, you can integrate the level of information that is coming to the cell at the level of transcription of a particular gene, in this case IL-2. So let us say I do not know exactly which signaling pathway it is. Let us say this is signaling pathway 1, this is signaling pathway 2. Okay, and this signaling pathway is activated if stimulus A is there, that might be bacterial infection. And this signaling pathway is activated if stimulus B is there, that might be inflammation. I don't know. I really do not know what are the options. But all I'm trying to say is that if both these instigations are there simultaneously, resulting in activation of signaling pathway 1 and signaling pathway 2, then only you will have both NFAT and AP1 in the same cell. And then they will be able to bind to the nucleus and activate transcription of IL-2. So either one of the upstream events, if activated, which might be the case in some other cell, like other DNA context, like where AP1 is needed in combination with something else, or just signaling one event has happened, NFAT is produced, that will not result in IL-2 transcription. It might result in transcription of something else, where some other combinatorial chemistry will be in action. But if signaling 1 and signaling 2 are happening simultaneously on a particular cell, then only NF80 and AP1 will come together and form this. Like, a parallel case you can think about is in the case of bone formation or cartilage formation. You do need BMP signaling and you need Wnt signaling. Both of these are needed. Then only you will get, like you do need some other signaling pathways as well. So we will come to those kind of complexities later. But the fact is that a particular cellular response, whether it is differentiation or mounting inflammatory response or whatever it is, or say blood coagulation, whatever it is the fail-safe mechanism is that just one instigation will not allow turning off on an entire pathway, but multiple instigations together will trigger the turning on of different of an entire pathway. Okay, uh, this is a slide that you really want to pay some attention to. So this is an enhancer, a very well studied enhancer for beta interferon. Again, another critical gene for your immune response. You see, it is only 50 base pair long. Okay, typical enhancer is 50 to 200 base pair. And see how many transcription factors are bound there. 1, 2, 3, 4, 5, 6, 7, 8. You see, like this is a very compact cluster of transcription factor binding sites. And these transcription factors, not only they interact with the DNA, they also physically interact with each other. And that's how this whole thing is recruited. So the take-home message is therefore that we talked about it earlier, that an enhancer is a cluster of transcription factor binding sites. Now, that particular enhancer will activate transcription of its target gene if, ideally, all of these transcription factors are present in the cell, or majority of them, 7 out of 8 or 6 out of 8 are present in the cell, then the transcription will happen. And these 8 transcription factors, production of each one of them can potentially be dependent on one kind of signaling pathway or the other. Okay, so therefore, at the level of this particular enhancer, multiple different signaling pathways are integrated, and then only this enhancer is turned on, active, and then beta interferon is transcribed. Okay, so this is something that I thought I should alert you about. That when you are thinking about transcriptional activation, you should think about enhancers just like as crowded transcription factor binding sites, and how the combination is important. Okay, just having 2 or 3 of them may not at all result in activation of transcription. You need all of them, presumably or ideally, to be cooperatively recruited to the DNA for transcriptional activation to happen. Okay, so therefore the take-home message from this particular lecture is that every transcription factor, you have a DNA binding domain and a transactivation domain. The DNA binding domain, they interact with the major groove of the DNA, primarily because of the basic amino acids and their typical signatures. Okay, like there are computer algorithms, and algorithms exist because there is a signature, right? You can feed any protein sequence and it will tell you whether this is likely to be a transcription factor because it can recognize the DNA binding domain. Okay, and then there are transactivation domain, which typically assumes a particular conformation upon DNA binding or upon the co-activator binding or upon the hormones for the nuclear hormone receptor factor binding, and then they are recruited together through protein-protein interaction as well as protein-DNA interaction for the enhancer to be active. Okay, so these kind of structures sometimes they are referred to as enhancosomes. Okay, fine. So that's all that I had to talk about on this particular topic. Then I will come back with another picture.